Welcome, everyone. Thank you for taking time out of your day today to join the Parkinson's Foundation's research webinar series. My name is Tom Manick, and I'm a passionate supporter of the Parkinson's Foundation's mission to make life better for people living with Parkinson's disease and their families. It is my pleasure to facilitate today's webinar to provide you with an inside look at the Foundation's Research Grants Program and the exceptional early career scientists working to uncover the fundamental clues essential to solving the Parkinson's puzzle. On today's webinar, President and CEO of the Parkinson's Foundation, John Lehr, will provide you with a foundation update. Dr. Jim Beck, Chief Scientific Officer, will highlight our research grants program, and Dr. Meghan Bucher will share an overview of her Parkinson's Foundation funded research. We will then wrap up the webinar with a Q&A session. Before we get started, I would like to review a few housekeeping items. This webinar will be recorded. A link to rewatch the webinar will be sent to you after the program, along with the slides and other resource materials. We have designated 15 minutes for the Q&A session at the end of the program. If you have any questions, please go ahead and submit them via the Q&A button at the bottom of your screen. If we don't have time to answer all of your questions, a Parkinson's Foundation staff member will follow up with you after the call. It's now my pleasure to turn it over to the President and CEO of the Parkinson's Foundation, John Lehr.
Thank you so much, Tom. I really appreciate it. And welcome everybody to this webinar. We're so pleased to have everyone joining us today. If we can go to the next slide. I just want to start with maybe a 30,000-foot view of who the foundation is. We are pleased to be the nation's first national organization covering the entire country. We were founded in 1957. At that time, we were two separate organizations, one in New York and one in Miami, and we've nicely merged to be the national organization, Parkinson's Foundation, in 2016. We've had tremendous growth since then, and that growth has allowed us to move from being just simply a granting organization to an organization that's really trying to drive change in the Parkinson's community, most notably around advancing research toward new and better therapies and ultimately a cure, but also being very mindful of helping people living with the disease today to improve their overall health and quality of life. So that everything that we do at the Parkinson's Foundation is really geared toward those two major areas of focus. Next slide, please. So the foundation has three core mission pillars, and I'll work from right to left just by starting with education. In education, the Parkinson's Foundation prides itself on being the go-to source for all things Parkinson's disease related. And we do that in many different ways. First and foremost, through our website, parkinson.org, which is rich in information and resources available on all subjects related to Parkinson's disease. We also do it through our toll-free helpline, 800-4PD-INFO, where you can call during working hours in the United States and reach a specialist in person, live, who has just an enormous, tremendous knowledge in terms of Parkinson's. In fact, I'll just make this point. One of our helpline specialists is soon retiring, and we did a calculation and determined that she has handled 16,000 calls over her tenure at the foundation, and she's just absolutely terrific. And that's true of all of the people who are on our helpline and will answer calls. We also do in-person educational meetings, webinars, podcasts. We're here to help people in their journey with Parkinson's and their care partners and their families, and to provide the things that need at the right time in the right way. Second is our mission pillars, care. And we are very much focused on improving care and the standard of care and the quality of care over time. Our biggest initiative this year is our Global Care Network, where we support the care of 64 centers of excellence. These are the leading academic medical centers, mostly here in the United States, but we have centers in Canada, Europe, and really all around the world. And the idea here is to support care, but also to learn what's going on in care so that we can, if there's a breakthrough in care or better treatment that comes along, we can disseminate that throughout our broad network and beyond our network. And we do that in many different ways, but most notably through our Parkinson's Foundation Conference, which is very well attended by leading clinicians and researchers across the world. And we have a lot of other initiatives in care that's really focused on, better models of care, ensuring that people who are going into the clinics are well prepared for their clinic visit with their clinicians. And more recently, we are launching an initiative to really invest more in clinical research programs. So care is a big component of ours. The last pillar, which I won't talk too much about because Dr. Jim Beck, who's our Chief Scientific Officer, is going to follow me and go into more detail. But we really have three main areas where we're investing in young investigators who have novel or innovative ideas in terms of basic research. One comment there is that Parkinson's disease is a complex disease, and there are many unknowns about Parkinson's. So we need a lot of great scientists researching the basic biology and the underlying causes of the disease. That's going to be the pathway forward to new and better therapies. We have a genetics initiative. We are one of the largest testers of people with Parkinson's for genetic mutations. We do that because we think that there are new therapies coming along targeting those genetic mutations, and that they may in fact be generalizable to the larger population. So people need to know their genetic status not only to enroll in clinical trials, but there may be some clinical reasons for that. And then lastly, we have a partnership with Parkinson's UK, which is the largest Parkinson's Foundation in Great Britain, where we are investing in early stage therapies targeted at controlling the symptoms of Parkinson's disease, and also, looking at disease modifying therapies. So that's our research portfolio. Again, I won't go into too much detail because Jim will talk about that in a minute, but it gives you a sense that the Parkinson's Foundation is moving on these and other fronts, including policy and advocacy. We are really here to help people with Parkinson's live their best lives. And we know it's going to take a lot of time and a lot of effort, a lot of support. And we're so grateful for all the people joining the call today to help us in that endeavor. So with that, I think I'm going to turn it over to Dr. Beck.
Thanks, John. I appreciate that introduction, and welcome everyone to our call today. I'm really excited to hear from Dr. Bucher about her research. So if we can go to the next slide, just to kind of recap what John was talking about when it comes to research, which is one of our main pillars at the organization. We break it up into three different approaches. You can think about it as a targeted investment horizons for those of you who think about their personal investments that way. So in the short term, we have our PD generation study. Our medium term is a virtual biotech that John talked about in combination with in partnership with the Parkinson's UK. And today what we're going to focus on is basic science investments. These are longer term investments that take a little longer to come to fruition, but very important as part of any diverse portfolio to have investments in these three areas. And so let me just take a little bit moment to kind of really underscore the hope for basic science and the importance of these investments as part of that process. We can do that in the next slide. So this is a headline that came from the Washington Post a couple of years ago, and it talks about cystic fibrosis. And cystic fibrosis, you may know, is a disease that affects children and now young adults and adults who deal with a condition that can make them more prone to lung infections. The Cystic Fibrosis Foundation invested heavily into basic science research, and the result was, this headline was a medication that, based upon understanding that basic biology, helps roughly 95% of people living with cystic fibrosis. So it turned what was really a fatal disease into a chronic disease, something that people can live well into adulthood and manage as part of that. And that's certainly where we want to go with Parkinson's disease. But I think it's important to understand when we talk about these advancements, they didn't happen overnight. And we can move to the next slide, please. So the moonshot was something that I think a lot of people have captivated in their mind about something that was, a great pronouncement came out from President Kennedy, and we were going to, move forward and get onto the moon in a short period of time. And sure enough, that happened. But the moonshot didn't just happen because of that. If we can go forward with just one build, they should happen automatically. I think it's important to recognize that there was a real history that led up to that moment where getting to the moon became an engineering problem that needed to be solved. And because there was that investments years in advance, we were able to take that knowledge and then apply it like an engineering solution in order to get us to the moon. But there really required investment along the way to really understand that process many years in advance. If we go to the next slide, I think we're not alone in recognizing, as we've always have been, that basic science is really critical to understanding a disease where we don't understand why it occurs, we don't understand why it progresses, and we certainly don't know how to stop it. So this is just a headline coming from Aligning Science Across Parkinson's. This is an initiative of Sergey Brin, who is a co-founder of Google, one of the few individuals who can write billion-dollar checks, and has, through his organization, ASAP, has really invested heavily following our lead that basic science is really the path forward to seeing real cures in our lifetime for Parkinson's disease. But, when we think about this is just the effort of one individual, but we have our federal government is in place in order to really help drive that forward. And the next slide just shows us one of the real challenges when we think about relying on just the federal government or others in order to do that. This graph from 2008, and I didn't update it since 2022, just shows in the black line spending for Alzheimer's disease research versus in the blue lines Parkinson's disease research. And you can see when the Alzheimer's Accountability Act passed, this is akin to our the Parkinson's National Plan that just recently got passed, there was a real change in the trajectory of funding for Alzheimer's disease research. And now we see there's about a 12-fold difference in the amount of money that's spent on Alzheimer's versus that in Parkinson's disease. And this is taking into account that there's about 5 times as many people with Alzheimer's as Parkinson's. So if you're a young researcher like Dr. Bucher is, and you're faced with this idea of a very competitive landscape on very difficult to receive funding, which path are you going to choose? Both are brain science, and both will certainly help people who are struggling with diseases. But we are the Parkinson's Foundation, and our focus is on Parkinson's disease, which is why in the next slide, the role of the Parkinson's Foundation plays is really here in de-risking Parkinson's research. Our goal here is to motivate scientists to devote their careers to Parkinson's research, whether they're starting off or they're making a switch midway. By giving them that incentive to kickstart their innovative ideas, we're hoping that through grants and fellowships and partnerships, we can really ensure we have the human capital that's necessary in order to really focus on Parkinson's disease as part of this effort. When you think about the research paradigm, researchers, and we can ask Dr. Bucher this when she speaks during our Q&A session, researchers don't do this as a 9-to-5 job. They are all in on this, and they spend many of their waking hours really thinking about the problems that face them, as well as maintaining their research moving forward. It requires going in on weekends, holidays. They are really devoted and almost in a monastic pursuit to trying to understand the problems that before them. And if we can, through our grants program, incentivize them to continue on that pathway, they can leverage our support in order to really bring in additional funding. And a 1-to-4 ratio is really quite amazing as part of that process. And the Parkinson's Foundation, in the next slide, is a source of funding for many people who are interested in doing Parkinson's research. And what we've seen over the years is that the amount of applications we receive has been increasing steadily. So this past year, we received 312 applications across these 5 different award programs that we have below. And you can see to the right of those numbers in bold are how many we have budgeted to be able to support. So roughly, of the 300 grant applications which come in, we're going to be able to fund less than 10%. And that's not to say that the remaining 90% are not good quality grants. It's just that we alone as an organization are limited in the amount of money that we have. So we have to draw a line based upon what we can fund. We could keep going down and continue funding grant after grant for a reasonable period of time and ensure that we have high quality research as part of the process. And this is where you support our community come into play in order to ensure that we're able to do that and hopefully be able to do that more moving forward as we talk about turbulent times for science today. So without further ado, I'd like to turn it over to Dr. Meghan Bucher, who is an associate research scientist at Columbia University and a past recipient of Parkinson's Foundation funding. Meghan?
Okay, well, hello everyone, and thank you so much for tuning in today to hear about the research that I do that has been made possible by the Parkinson's Foundation. And thank you for that introduction. As Dr. Beck mentioned, my name is Meghan Bucher. I'm an associate research scientist at Columbia University, working in the laboratory of Gary Miller. The Parkinson's Foundation supported my work as a postdoctoral research fellow from 2022 to 2024. And this is on a project titled Therapeutic Targeting of SV2C in Parkinson's Disease. And before I talk about my research, I wanted to start just with a quick introduction to myself. I've been researching Parkinson's disease for over 10 years. When I first started studying neuroscience, I got really interested in the fact that neurons are unlike any other type of cell in the body. They don't regenerate, so when neurons die, they're gone for good. I wanted to understand both what could cause neurons to die and also what mechanisms might be able to prevent neurons from dying. So this led to my research as an undergraduate studying antioxidant proteins that our bodies produce. These are called selenoproteins. And this was to see how selenoproteins can protect against oxidative stress and neurodegeneration in a rodent model of Parkinson's disease. When I started graduate school, I switched from studying these protective antioxidant proteins to studying causes of oxidative stress in Parkinson's disease. So this work investigated how dopamine, which is a neurotransmitter that I'm sure you've all heard of, may have the potential to induce neurodegeneration simply through the mismanagement of its packaging. And so we found that disrupting the packaging of dopamine in rats was sufficient to cause neurodegeneration that mimics what is seen in Parkinson's disease. After I received my PhD, I moved to Columbia University as a postdoctoral research scientist, where I continue to work as an associate research scientist, studying ways we can therapeutically target dopamine regulation to protect dopamine neurons from degeneration in Parkinson's disease. To provide a bit of context for the work that I do, dopamine is responsible for a lot of things in the brain, but one important role of dopamine, especially for Parkinson's disease, is to facilitate movement. So dopamine pathways consist of neurons that use dopamine to communicate with each other in a process called neurotransmission. In order for a neuron to use dopamine as a neurotransmitter, there is a sophisticated system of regulation that involves the production, storage, and release of dopamine. So as I progress through my slides, we're going to zoom in on this neuronal pathway. First, we'll look at this area called the dopamine synapse that exists at the intersection of where these two neurons in the pathway are communicating. So at the dopamine synapse, we have our first neuron in the pathway. This is our dopamine neuron, and it releases dopamine to communicate with the second neuron in the pathway. So this is our dopamine receiving neuron. In the dopamine neuron, dopamine is synthesized in the cellular space that we call the cytosol through this pathway. Once dopamine is synthesized, it needs to be packaged into synaptic vesicles. Synaptic vesicles that are filled with dopamine will release their contents to communicate to the next neuron in the pathway, in this case, to facilitate motor behaviors. The released dopamine can act here on the second neuron, and then once it has its intended effect, dopamine can be taken back up by the releasing neuron to be repackaged for future use or broken down. Each of these processes are necessary for functional dopamine neurotransmission. When these processes aren't working or the dopamine neurons degenerate, there is a deficiency in dopamine communication occurring here, and this is what causes the motor symptoms of Parkinson's disease. So you'll notice on the diagram here, L-DOPA. It is the precursor to dopamine. It's also the most common therapeutic to treat Parkinson's disease symptoms. And L-DOPA works by increasing the amount of dopamine that can be released during neurotransmission to restore this communication and help alleviate motor symptoms. So as we continue to zoom in, now taking a closer look just at the synaptic vesicle, this is the necessary step after dopamine has been synthesized, where it's packaged into the vesicle to be ready for release. So at the synaptic vesicle, there are two key proteins that I'm going to be talking about today. These are the vesicular monoamine transporter 2, or VMAT2, and synaptic vesicle glycoprotein 2C, or SV2C. To show you how VMAT2 and SV2C act, we can look at the synaptic vesicle. And here there's a protein called V-type ATPase that is responsible for packaging protons, or these hydrogen ions, into the vesicle. VMAT2, which is our dopamine packaging protein, can then exchange these protons for molecules of dopamine. So this results in a movement of protons out of the vesicle into the cytosol, and movement of molecules of dopamine into the vesicle and out of the cytosol. While the exact function of SV2C is unknown, our previous work has suggested it may play a role in enhancing the amount of dopamine in the vesicle and retaining the dopamine in the vesicle. So I've depicted this here as the SV2C kind of holding on to the dopamine molecules, which frees up space for VMAT2 to load additional dopamine into the vesicle. And when SV2C holds on to dopamine, it's also keeping the dopamine in the vesicles. So we have these two proteins that are working in concert. We have VMAT2, which is packaging dopamine into the vesicle, and SV2C, which is increasing how much dopamine can be in the vesicle and helps keep the dopamine inside the vesicle. These processes are incredibly important because unpackaged dopamine that remains in the cytosol can be neurotoxic. So for example, if VMAT2 is unable to package dopamine into the vesicle, this results in an accumulation of dopamine out in the cytosol, where it will undergo neurotoxic reactions, generate oxidative stress, and this can cause neurons to degenerate. So just to summarize, the packaging of dopamine into synaptic vesicles. This is important for regulating motor function, so factors like VMAT2 and SV2C that keep the vesicles filled with dopamine can help mediate motor function to make sure dopamine neurotransmission is occurring properly. And the dopamine packaging in the vesicle is also important for keeping the neurons healthy, because factors like VMAT2 and SV2C keep dopamine out of the cytosol, which means there will be better neuroprotection. There is evidence that VMAT2 is impacted in Parkinson's disease. There are some rare gene variants that have been identified that either increase VMAT2 expression or decrease VMAT2 activity, and these variants are protective or contribute to Parkinson's disease risk, respectively. This makes sense if you think about VMAT2 as being a protective mechanism where it's keeping dopamine safe within the vesicle and out of the cytosol where it can be cytotoxic. The more VMAT2 function you have, the less cytosolic dopamine and the healthier neurons. Beyond genetic evidence, there is also significant evidence to suggest that VMAT2 function and expression are impaired in Parkinson's disease, regardless of whether you have a gene variant in VMAT2, which suggests it may be a common pathway that's impacted in all cases of Parkinson's disease. SV2C is very similar to VMAT2, and that rare gene variants in SV2C have been implicated in Parkinson's disease, with some variants increasing Parkinson's disease risk and some variants decreasing Parkinson's disease risk. There are also some variants in SV2C that modify aspects of Parkinson's disease, such as disease severity, the age of onset, or patient response to medication. And I want to highlight this last study because it found that SV2C variants can reduce the amount of L-DOPA required in order to see symptom relief in people with Parkinson's disease. This suggests that a therapeutic that could target SV2C could help in symptom management for people with Parkinson's disease. Just like with VMAT2, beyond genetic evidence, there is significant data to suggest SV2C function is impaired in Parkinson's disease, regardless of whether you have a gene variant in SV2C, which suggests it may also be a common pathway impacted in all cases of Parkinson's disease. To bring all of this together, we know that genetic and environmental factors can contribute to Parkinson's disease risk. And in Parkinson's disease, there's deficient dopamine neurotransmission due to neuron dysfunction and neurodegeneration. And this deficiency in dopamine transmission causes motor symptoms, but dopamine replenishing therapies such as L-DOPA can help treat the motor symptoms in Parkinson's disease. So what I'm really interested in is whether we can directly target the vesicular regulation of dopamine as a therapeutic intervention by designing drugs that act on VMAT2 or SV2C, which can help preserve dopamine neurotransmission, potentially as a co-therapy with L-DOPA, which could also help protect against neurodegeneration and Parkinson's disease and help with motor symptom onset. And so to quickly comment on the parallel approaches of targeting VMAT2 and SV2C, so far I've been talking about VMAT2 in the context of dopamine, but what I haven't mentioned is that VMAT2 is also expressed in multiple neuronal populations. So it's also responsible for the vesicular packaging of other neurotransmitters, including serotonin, histamine, and epinephrine. In comparison, SV2C expression is enriched specifically in dopamine neurons. And SV2C is part of a family of 3 related proteins. These are SV2A, SV2B, and SV2C. And what I'm showing here are mouse brains viewed from this side that have been cut in half down the middle to reveal the internal brain structures. And these brains have been stained to visualize the 3 SV2 proteins. What you see for SV2A is this widespread dark staining that shows that it is widely expressed throughout the brain. In comparison, SV2B has much less expression, and SV2C has these specific brain areas that show dark enriched expression, and these areas consist of dopamine neurons. So while we think that both VMAT2 and SV2C are good targets for therapeutic intervention, it's possible that targeting SV2C may have a more specific benefit because it would only be affecting dopamine systems. So before I talk about our work on SV2C, I did also want to highlight some of our work on VMAT2 that has also been funded by the Parkinson's Foundation. And this was work for an undergraduate student who works under my mentorship, Ava Knickerbocker. Ava received a summer student research fellowship last summer, which supported full-time research in the lab. Ava works on VMAT2 targeted therapeutics, and actually just this morning presented her work at an international science conference, the Society of Toxicology meeting. And her attendance of this meeting has also been made possible by the support of the Parkinson's Foundation. So back to SV2C. I hope by now I have been able to convince you that SV2C is a viable therapeutic target for Parkinson's disease that may modify disease progression by protecting against dopamine toxicity and improve the efficacy of symptomatic interventions. The work that I performed during my fellowship at the Parkinson's Foundation was an effort to identify or design a drug that would positively influence SV2C. I took a dual approach for this project, and this was first by establishing a cell-based model that allows us to have subcellular resolution directly looking at the synaptic vesicle to evaluate whether drugs can influence vesicular dynamics. So when we study cells in culture, it allows for a very controlled and isolated system. In our case, we use cells that are derived from human embryonic kidney, or HEK, that we have engineered to express VMAT2 and SV2C. So here I'm showing the validation we performed on the cells that we engineered to confirm the expression of both of these proteins by labeling them with a fluorescent marker visible during microscopy. We have one cell line that only expresses VMAT2. So you see the VMAT2 protein here without SV2C, and another cell line that expresses both VMAT2 and SV2C. In our cell line that expresses both proteins, we can take a closer magnified look at individual cells, and we see that there's co-localization of VMAT2 and SV2C on these subcellular vesicles. So despite these cells being in a dish and originating from the kidney, they are mimicking what we see in neurons, where these 2 proteins are expressed on vesicles together. To study the function of VMAT2 and SV2C, we can use a fluorescent false neurotransmitter, or FFN, that has been designed to mimic the structure of dopamine. So this is essentially a fluorescent dopamine that we can use. So here I'm showing the structure of dopamine and the structure of the particular FFN I use, FFN206, with their similar structures. And because FFN206 looks like dopamine, our dopamine packaging protein, VMAT2, will package it into vesicles. This compound possesses a fluorescent property, which allows it to be visualized and quantified using microscopy. So what I'm showing here on the right side of my slide is a video taken of our cells with the FFN packaged into vesicles inside them. And so you can even see in some of these areas that over time, there's this trafficking or movement of the FFN-filled vesicles throughout the cells. Because the FFN allows us to visualize the activity at the vesicles in the cells, we are able to use this system to investigate how SV2C impacts vesicular dynamics. When we put FFN 206 on the cells, over time, it gets packaged into the subcellular vesicles. So we can measure how much FFN is in the vesicle by looking at how much fluorescence there is in the cells. And so what we saw was that in our cell line that expresses both VMAT2 and SV2C, there is an increase in the amount of FFN that is packaged into the vesicles by about 33%. This is consistent with our hypothesis that SV2C is helping to enhance the uptake of dopamine, or in this case, FFN, into the vesicles. We can also investigate the other part of our hypothesis that SV2C holds on to the dopamine or the FFN to help keep it in the vesicles. So in this experiment, we allow the vesicles to fill with FFN and then measure how much FFN stays in the vesicles over time. We think that this is an active process. So over time, some of these molecules of FFN may leak out of the vesicle, but then they can be taken back up into the vesicle by VMAT2. However, over time, some of the FFN signal degrades, likely due to breakdown of the FFN. When we measure the amount of FFN in the vesicles over time, we see that the cells that express both VMAT2 and SV2C have a slight protection against this loss of fluorescence over time compared to the cells that only express VMAT2. So that's this top line here. But this is measuring both the leakage and the reloading of FFN over time. So to isolate just the retention component of this process, we can modify the assay and use a drug that is inhibiting VMAT2. So this will prevent the reloading of FFN into the vesicles over time. In this experiment, the FFN leaks out and it is not able to be packaged back into the vesicles. So it is simply a measure of retention or leakage of FFN. We see a much greater separation between the two cell lines here, where the cells that express both VMAT2 and SV2C retain the FFN in the vesicles much better compared to the cells that only express VMAT2. This is really significant evidence that SV2C is helping to hold on to the FFN in the vesicles, which supports our hypothesis that SV2C has a role in the retention of dopamine in vesicles. Now that we have these cell culture assays established, we can test whether SV2C therapeutics further influence SV2C's effect on either total FFN uptake or the retention of FFN within vesicles over time. To complement the work that we do in cell culture, I also wanted to test compounds in a whole organism. So I established a model using the model organism C. elegans, which is an invertebrate nematode, or basically a microscopic worm. The C. elegans model allows us to test the drugs in an organism for effects on motor behavior and neuron health, which will provide a more direct insight into how these drugs may influence Parkinson's disease outcomes in humans. As I mentioned, C. elegans are microscopic nematode worms. They are transparent and about 1 millimeter in length when they are fully grown. So here I'm showing a microscope image of one of these worms. Despite being an invertebrate animal, they have conserved dopamine biology compared to humans. So they have 6 dopamine neurons in their head, which I'm showing here in this image, where the dopamine neurons have been labeled with a fluorescent protein. This allows us to visualize them using microscopy, which is a very useful tool because it allows us to evaluate the dopamine neuron health by easily looking at the dopamine neurons. So I mentioned that C. elegans have conserved dopamine biology compared to humans. So just like in humans, many of the motor behaviors in C. elegans are also mediated by dopamine. So we can record videos of C. elegans as they swim or crawl to look at their motor behavior with measures such as thrashing, which is this kind of body bending movement as they swim, or curling, which this worm on top is doing a lot in this video. And during the crawling behavior, we can look at things like speed, how quickly they're moving, or the distance they travel in a given time period, just as measures of their mobility. So studying dopamine in C. elegans, as I showed in the previous slide, we can visualize their dopamine neurons using fluorescence, and we're able to measure their motor behavior through these swimming and crawling assays. While C. elegans already express a similar protein to VMAT2, that allows for vesicular packaging of dopamine, they do not naturally express SV2C. So we had to create a new strain of C. elegans through genetic engineering to express SV2C in the dopamine neurons. So we can visualize this through microscopy. Here I'm showing the head of one of these transgenic C. elegans. And the SV2C protein in the dopamine neuron is labeled with a pink fluorescent marker. And these worms also have this green fluorescent marker in their pharynx, and that is just a larger marker for us to visualize to show the introduction of the SV2C protein. So now we have a model of C. elegans that expresses SV2C, but in order to evaluate how SV2C impacts neuron health, I needed to actually create another strain of worm. And so what we did is we took our SV2C expressing animal and crossed it with another strain of C. elegans that expresses a fluorescent protein in its dopamine neurons. So now we have the bottom here, a new strain of C. elegans that not only expresses SV2C in the dopamine neurons, but also GFP or fluorescent protein that is allowing us to visualize the dopamine neurons to evaluate their neuron health. And we can also use these worms to measure motor behavior, such as distance crawled as a measure of their mobility. So when we take a video of the worms crawling, we can use an automated software that is measuring how far they move in a given time period. So here you're seeing each green area is an individual worm that's been recorded in a video, and the red line is the track of where it has moved across the video. And so what we first did was test whether SV2C could protect against motor deficits in a model of Parkinson's disease using a dopamine toxicant called MPP+. So MPP+ is often used to model Parkinson's disease because it causes dopamine neurons to degenerate. And in C. elegans, when dopamine neurons degenerate, we see less movement. And so what we were able to determine was that in worms that do not express SV2C, when they are exposed to MPP+, they travel a shorter distance, about 35.6% decrease in the amount of distance traveled. However, in the worms that express SV2C that were exposed to MPP+, they only have a decrease in distance traveled about 22%. So this suggests that the SV2C was able to protect the dopamine neurons from the toxicant MPP+ and helped to preserve motor behavior. I also mentioned earlier that we believe SV2C may be able to make L-DOPA treatment more effective in alleviating motor symptoms in Parkinson's disease. So to study this, we have also used a strain of C. elegans that overproduces dopamine, which models L-DOPA supplementation. So in C. elegans, dopamine not only controls motor behavior, but it also determines body size. And we see that the L-DOPA model strain that overproduces dopamine has a decrease in body size as the worms develop through their larval stages into adulthood. This is this bottom line here. And this is compared to our SV2C expressing worm on the top here. When I bred these two strains to generate a worm that overproduces dopamine and expresses SV2C, we saw a rescue in body size. So this suggests that deficits that are caused by the overproduction of dopamine can be rescued by SV2C, supporting the idea that SV2C can help regulate increases in dopamine production or improve the effectiveness of L-DOPA. So now we have cell culture assays that allow for subcellular visualization of vesicular dynamics and multiple models of SV2C function in C. elegans. So now we need therapeutic compounds to test in these systems. We are very fortunate that there is precedent for successful therapeutic targeting of SV2 proteins. There is a widely prescribed and well-tolerated medication called Keppra, which is used to treat epilepsy and acts at SV2A. So as a reminder, SV2A is one of the related proteins to SV2C, and it has more widespread expression throughout the brain. While Keppra acts at SV2A to help manage epilepsy and seizures, it also has an interaction with SV2C. So what we would ideally like is to have a drug that has a higher selectivity and specificity to SV2C over SV2A and SV2B to make sure the drug is specifically acting at the dopamine neurons where SV2C is expressed. And so we can use theoretical modeling of SV2C's protein structure, which I'm showing here, to visualize how SV2C interacts with Keppra And this visualization allows us to make modifications to the drug structure to see if we can design a version of this drug that has better interaction with SV2C. We've used these techniques to design new compound structures and measured how well they interact with SV2C. So in this table here, I'm showing 31 new compounds that we tested, where the larger the score, the better they interact with SV2C. So to conclude everything I've shown you today, the work I performed while funded by the Parkinson's Foundation allowed us to develop these cell culture and C. elegans models to test therapeutic compounds that act at VMAT2 or SV2C. So now we're at a point where we can use drug discovery tools to design these new therapeutics to test. We can test them in our assays. We can look at the effect of these drugs directly at the vesicle with our cell culture experiments to measure the effect they're having on vesicular dopamine dynamics, but we can also test these compounds in our complementary C. elegans model to determine how the drugs improve neuron health, motor behavior, and L-DOPA treatment for more direct Parkinson's disease relevance. And I wanted to take a minute to mention an award our group was recently received from a new organization called Spark-NS, which is funding our continued work on developing therapeutics that prevent dopamine-induced neurotoxicity in Parkinson's disease. Before the Parkinson's Foundation funded my postdoc fellowship, we submitted several grant applications for this work, none of which were funded. When the Parkinson's Foundation chose to invest in me and the development of this project, I was able to generate additional data and evidence and tools that made it possible to apply for and receive this future funding. This award that we received from Spark-NS is a $2 million award with a specific mechanism to help academic scientists develop their findings beyond the lab and into clinical settings. Too often, the findings we make in an academic setting are not able to translate into actual therapeutics available to patients because academic scientists are not equipped with the tools or the expertise to make that transition happen. Through Spark-NS, we are not only receiving funding to support our research, but we have also been connected with an entire team of industry experts who are helping to support us and advise the development of our research project. I also wanted to mention, I saw some questions coming in through the Q&A. You may have heard in the news recently that academic institutions, and specifically Columbia, have experienced recent cuts to federal funding. This supports a lot of important research. And so we are particularly grateful for private foundations like Parkinson's Foundation and Spark-NS, whose support is allowing our work to continue in this otherwise very uncertain funding climate. And so with that, I would like to thank you all again for taking the time to tune in and listen to my presentation today. I would like to also acknowledge the members of the Miller Lab who have helped contribute to this work, as well as our collaborators and especially our funders who have made all of this work possible. And I would also be happy to take any questions.
Thanks so much, Megan, Jim, and John. The work being done through the Foundation's grants program is incredibly inspiring and certainly provides hope for those living with Parkinson's and their families. And we can see with what Megan's been doing, it will help in the future. Parkinson's is very personal for me, as my wife, Ro, lived with the disease for over 30 years. I've been involved with the Parkinson's Foundation in many capacities, as a volunteer and as a donor. Supporting the Foundation's research initiatives, like the grants program, is a priority of mine. I take pride in knowing that through my contributions, including our planned estate gift to the Foundation, I'm helping to fund innovative studies, the brightest researchers, and new programs to identify promising new treatments and ultimately a cure for Parkinson's. I will now turn it over to Jim, who will facilitate the Q&A session.
Thanks, Tom. I appreciate that. Megan, thank you very much for what a great talk and really interesting research that you have completed as part of that. Several questions have come in, and just want to just ask one of them. You talked a little bit about your research and how you got started. What got you into neuroscience to begin with? I mean, what keeps you going as part of the process? Because as I mentioned, there are a lot of opportunities. You're a very intelligent person, well accomplished. What keeps you focused on Parkinson's disease?
Sure. So I feel like I came into science a little bit late, I think, for most people compared to most people. I went to a boarding school for the arts in high school. I didn't know that neuroscience existed before I got to college. I was generally interested in psychology. When I got to college, I took a class called Brain Mechanisms, thinking it would be related to psychology, and it was actually a neuroscience course. And it kind of opened my eyes to the world of neuroscience. I didn't know that you could even study brains at that level. So that first class that I took, it completely changed my trajectory, and I never looked back, and I've been committed to neuroscience research ever since. Um, I can't imagine doing anything else with my life at this point. I think being a scientist is the best job in the world, and it is so personally fulfilling. I love that I get to wake up every day and go into the lab and ask questions and design experiments to try to get at these answers to better understand how we all work. And it's also really gratifying to work with young students who are getting their first experiences in research and be able to pass that knowledge on to the future generation. So I think what keeps me going is often the interactions I get to have with other scientists, but I truly can't imagine doing anything else with my life.
Yeah, it sounds amazing, especially what you've accomplished and that focus. It's what an interesting shift from the arts into really hard science as part of the process. One of the things I think is also helpful too, because I really appreciate the kind of the background and how you got to where you are today as part of the process. There's a lot of people you had on your acknowledgement slide as part of the process, but I think it's important to recognize you're doing a lot of the work yourself, I imagine, as well. So who keeps your worms alive? Who keeps your cells alive? I mean, what's going on as part of your day, your work week in the lab?
Sure. So I am still very, very hands-on with my research. You mentioned at the beginning of the webinar, that this is not a typical 9 to 5. And I kind of internally laughed because over the past, I think, 4, 5 straight weekends I've been in on Saturdays and Sundays to keep my worms alive. So this is it is not a typical job, but it's something that is very fulfilling, and it helps to keep it manageable. But I am very hands-on with my research. I do have a team of 3 undergraduate researchers who work under me. So we work together. I help them set up their experiments, and they are becoming extremely independent, well-rounded scientists on their own. And so it is definitely a team effort. We have many members of our lab are always helping to each other on their projects. But it is something that I am very, very involved with still, hands-on. I spend all of my time at the bench actually performing all of the experiments myself.
Yeah, fantastic. So it definitely keeps you busy, but it also probably gives you the insights to where successes and failures may come from as well as part of the process. So speaking of your research, one of the things I was thinking about is you were talking about this protein SVC2, and it's in this vesicle, and it kind of acts like the way you described it, almost like a little sponge, where it absorbs the extra dopamine in the vesicle and help it maintain, so you can just pack more. The VMAT can pack more of the dopamine in there. But what's interesting is you talked about these FFNs, these false fluorescent compounds. Are they close enough to dopamine to mimic that sponge effect? Because it was interesting to see that even with this synthetic compound that's not dopamine, you were able to measure and see effects from your experiments to show that the presence of SVC2 could help pack this fake, I don't want to say fake, I guess it's false, compound in there, and then you could then perhaps extrapolate the benefits for with dopamine. What are your thoughts on the ability of that to kind of really replicate real dopamine?
Yeah, so I didn't include all of the validation experiments we did just in the interest of time today, but when we were first establishing these models, we also did a comparative study with actual dopamine itself, and also with vesicles that were derived from the brain of mice. And so what we did was kind of mimic the FFN experiments, either using a radio-labeled dopamine or a radioactive dopamine. We can measure its radioactivity. And we did that either in the vesicles from the cells or vesicles from the mouse brain. And what we found across all of those experiments was that regardless of whether the vesicles were derived from a mouse brain or from cells, if they were using radiolabeled dopamine or FFN, they all showed the same results. And so this was actually very helpful for us because it is a much easier system to do these drug testing experiments in cell culture if you can use this fluorescent dye that makes it actually possible to visualize the dynamics. You don't have to expose yourself to radioactivity. You don't have to use any animals. It makes it a much more amenable to higher throughput testing. So I think that our data, when we compared these results from with the animal results and the dopamine results, really helped us to validate our system, and that we feel very confident that what we're seeing with the FFN is replicating what we would actually expect to be happening in the brain.
Fantastic. So, and those changes you showed, they looked subtle on the graph. I mean, you were only doing it maybe, it was 60 minutes was the length of time. So, what do you think the impact will be, on either on the animals, or if you were to take it to mice, or hopefully one day it's successful in treating people? Do you think you only need small changes? I mean, what can you share about the amount of dopamine and the changes you could see with this potentially getting a deeper understanding of this compound, this molecule?
Yeah, I think that even a small change, a small increase in uptake or the retention can have really large effects long term, especially if we think about Parkinson's disease as being a progressive disease. And so if this is something where you could start taking a drug that is making your vesicles better at packaging dopamine and releasing dopamine very early in the disease, it may help you to stave off how long it takes before you need to start taking L-DOPA, or when you start taking L-DOPA, it could reduce the amount of L-DOPA you need to see symptom alleviation. And I think that because we know that the cytosolic dopamine can contribute to some neurodegenerative processes, if you have anything that is helping to keep that dopamine in the vesicle, it's going to potentially slow the progression of the disease. So if you are making the neurons happy and healthier by targeting SV2C or VMAT2, it may have a small effect on the day-to-day or on the individual vesicle, but over time, it would be helping to not only improve your dopamine transmission for motor facilitation, but also keeping the neurons healthy.
Yeah, of course, that's fantastic. And we can only hope that we'll get to that point. So questions come in from the Q&A and just asking about how you share your results and coordinate your research with others. They mentioned Fox Foundation, but science is not just Parkinson's Foundation and Fox Foundation. There's a world of scientists out there. How are you, getting word out about what you're doing to the rest of the scientific community?
Yeah, so we have a lot of established collaborations with other labs, either at Columbia or other universities that we're meeting with on a weekly basis to talk about our experiments and get feedback from other experts. Some of these can be more casual conversations or more formal presentations. And then on a larger scale, I'm going to conferences on a very frequent and regular basis, at least 2 to 3 a year, presenting my work to a variety of audiences. I think it's good to have feedback from specific Parkinson's audiences, but also more broad neuroscience audiences, because you can get a lot of insight from people who are coming from different backgrounds and approaching it from different perspectives. And so as I mentioned, I had a couple of my students attending the Society of Toxicology meeting this morning. There is a large section of that meeting dedicated to studying dopamine toxicity. So I'm eager to hear what the feedback they got from their poster presentations today from some toxicologists. But it's just one example of the type of conference that we can use, where we go into, we present our work, and it's always in progress to get feedback to try to help figure out the best way to do the experiments. How can we improve what we're already doing, make connections with other scientists, and maybe they have existing tools that we could use and we could collaborate on that could help really develop and progress the project.
Yeah, for sure. Another question's come in asking about the dopamine toxicity. So why is intracellular dopamine toxic, and does that toxicity kind of leak out and then impact the neurons around it?
That's a very good question. So, this is kind of a really interesting feature of dopamine itself, where its chemical structure just lends itself to participating in these really neurotoxic reactions. And to get a little scientific here, it has to do with the pH or the acidity of the environment where it is. And so, in the vesicles, this is an acidic environment and it helps to keep dopamine very, very stable, but it's also sequestered away from everything else in the neuron. When the dopamine is not in the vesicle and it's out in the cytosol, this is a less acidic environment. And it's just a very reactive environment for the dopamine. It will immediately start to undergo these reactions that are creating reactive metabolites. They attack proteins, they attack lipids, they can attack DNA, they can interact with other proteins, cause downstream dysfunction, and just kind of render a state of chaos in the neuron. And so it is just to do simply with the structure of dopamine. And so that's why it's so important to have it sequestered away in that vesicle where it's stable because of the acidity. And it's also just away from anything that it would even attack. It's a really interesting question to know how that may spread to surrounding neurons. I don't think we have as much understanding because that process of dopamine release and reuptake and repackaging is so elegant in neurons. It's all very protective of the neurons. It's evolved that way. So when you have dopamine release into the synapse, it communicates with the next neuron, and then it gets taken back up by the first neuron. It is immediately packaged away into a vesicle again to be recycled for future use. So the neurons are very good at keeping dopamine in a safe location in the vesicle. And so I think in a normal healthy functioning neuron, and when those systems are happening the way that they should, there's not too much concern about how that could be spreading to the surrounding neurons. But when these systems start to break down, it could definitely have effects on the other neurons surrounding it.
Yeah, I guess it's good to be thinking that our cells are really chemical factories, and
Yeah.
when a chemical factory has a spill or something goes wrong, it can really mess up the environment around it for sure. Meghan Bucher, I want to thank you for your time. It was a great presentation. Really appreciate everyone who's able to join us. And I think I turn it back over to Tom, and you can close it out for us.
Thanks, Jim. And really, like you said, thank you, Megan, for everything. But for all the people who are watching this webinar, thank you very much for joining us today. We are incredibly grateful for your continued support and support of the entire Parkinson's community. We look forward to sharing more updates on the Foundation's research priorities and future programs. I urge each of you to join me in supporting the Parkinson's Foundation research initiatives to help bring us closer to a world without PD. If you have any questions about today's webinar or would like to discuss ways to support the program, please remember that the Parkinson's Foundation staff is always available to help. You can reach out to your staff contact or Kelly Austin at kaustin@parkinson.org. Again, thank you very much for being here.